Okay, so we can start. Welcome everyone to today's edition of Tissue Talks. I would like to remind you to please post your questions using the Q&A feature, which is at the bottom right of your screen. And then our speaker will address as many questions as possible after her talk. I'm really delighted to welcome today's speaker, Professor Elaine Fuchs from the Rockefeller University, who is a real legend in our field. Dr. Fuchs received his bachelor's degree in chemistry from the University of Illinois, Champaign-Urbana, and she went on to Princeton University to obtain her PhD in biochemistry. After that, she moved to Boston to postdoc at MIT with another legend, Howard Green. And this is actually where I believe she discovered her passion for skin and skin-related research. She was recruited to the University of Chicago in 1980, where she progressed through the ranks from assistant professor to associate professor and full professor. And in 2002, she moved from University of Chicago to the Rockefeller University, where she still here as chaired professor, Rebecca Lansfield Professor of Mammalian Cell Biology and Development. During the time before coming to Rockefeller, she also obtained a very prestigious appointment of the Howard Hughes Medical Institute investigator. Her research is really unique. Her laboratory is using mammalian skin as a model for research to study the remarkable properties of the adult stem cells residing in skin. Her team is interested to find out how do the cells know what to do and when. So it's about the selection of task and selection of timing. She is very much focused on the reciprocal crosstalk between the stem cells and their tissue environment in the context of sensing and communication. And again, all of this in the context of development, aging, inflammation, and cancer. Her lab is the one that discovered that epithelial stem cells can acquire and maintain their epigenetic memory, and that this memory can be both beneficial, for example, by enhancing wound healing, and maladaptive, for example, by causing chronic inflammation and susceptibility to cancer. Dr. Fuchs believes that the underlying mechanisms of these complex events can be identified and can be used to guide the therapeutic discovery. Her lab found that the cancer cells can hijack the mechanisms by which stem cells replenish dying cells and repair wounds. Her publication record is incredible. She published very many papers in top journals, in particular in Science, Cell, and Nature. And as many of these papers are really establishing the standards in the field and direction of our research. She has too many awards to list. I will just mention a few. She is a member of the National Academy of Sciences, National Academy of Medicine, American Academy of Arts and Sciences, American Philosophical Society, and the Royal Society. She obtained the White House Outstanding Scientist Award. She obtained an award from Columbia in 2002. This is Cartwright Award. L'Oreal UNESCO Award in 2010, National Medal of Science, Lifetime Achievement Award from the American Skin Association, and Benjamin Franklin Medal in Life Science. And I would like just to say at the end that I know from her many trainees that she is a passionate and caring mentor and exceptionally successful mentor, really dedicated to train the next generation of scientists. And let me close with a citation with one sentence that I pulled out from one of her many interviews. She says, "The more I work on becoming a better scientist, better mentor and participant in our scientific community, the richer all aspects of my life become. Elaine, thank you so much for being with us today, and you can now take over the screen and start your presentation.
Great. So let me just pull up my presentation here. And can everyone see that? Let's see, I guess I need to hit this.
Perfect.
All is good. Okay, Gordana, thank you very much for the invitation to participate in this Tissue Talk series. It's a wonderful concept and a great idea, and so glad to see this extended well beyond the COVID pandemic days. So today I thought I would tell you a little bit about some of the work that Gordana alluded to with regards to stem cells, how stem cells remember experiences that impact tissue fitness. We work on adult stem cells of the skin, and I always like to start my audience with some photographs of recent trips that I've taken. The photographer is me. In another lifetime, I'd love to be a photographer. But for today, I use this as an example to illustrate that there's no other organ of our body that displays this kind of diversity in evolution in order to be able to cope with the extremes of our environment. And I just think there's a treasure trove of fascinating questions that one can ask about the skin. And I think we naturally have an interest in this tissue as our body's surface. And so why should we use skin as a research model for studying stem cells? Gordana alluded to a few of these aspects. First of all, these were the first stem cells that were ever cultured, and they were among the first to be used in organoid cultures. And I'm just going to see, should I hide this or do anything here to remove the, I guess I
No, it's fine.
Everything is clear to you? Okay.
Yes.
Okay. And the epithelial stem cells of our skin are accessible, they're abundant. And for the mouse hair follicle, they are synchronous. The hairs are either rapidly regenerating and producing hair, or they are sitting there at rest, sometimes for very long periods of time. All of these tissues that you see here in blue, the epidermis, the outermost layer of which is the surface of our skin, the sweat gland, the sebaceous gland, the hair follicle, they're all derived from a common embryonic progenitor that expresses a set of keratins that I had been working on back when I was a postdoctoral fellow. During homeostasis, or the equilibrium of the tissue, the skin stem cells continually regenerate the body's surface, and after injury, they repair it. And then finally, more than any tissue of our body, our skin is exposed to a myriad of stresses that the stem cells must cope with. And so, as Gordana alluded to, back when I was actually a graduate student at that time, I heard a talk by then Howard Green, who had just developed a method of taking human skin and putting it into a tissue culture dish and being able to generate from it cells that could endlessly divide and still make skin. I was fascinated by this as a model system. It turned out that the breakthrough was the realization that stem cells rely upon other cell types for their sustenance. And so this turned out to be the foundation for embryonic stem cells, which were then cultured in the 1980s. Howard Green was developing this methodology for burn operations. When I started my laboratory, I was very interested in setting up three-dimensional organoid cultures to be able to recreate human skin in a dish. So from Howard Green's applications, this turned out to be a long-standing, successful use of stem cells in a clinical setting, where all was necessary was to take a small area of the unaffected area of a burn victim, place the cells into culture, expand them, and then graft them back onto the burned area. And this turned out to be very successful. what we learned from these studies is that only a few stem cells are necessary for whole body replacement. I think this is just remarkable. And that really turned out to open the door for many, many studies on stem cell research. When I was starting my own laboratory, I focused on the major proteins that were expressed by epidermal stem cells. In fact, I began that when I was a postdoc at MIT. I had shown that they are constituting a large gene family. They're broadly and differentially expressed in different tissues. And then when I began at the University of Chicago, it was at the cusp of DNA recombinant technology. And so we cloned and characterized the epidermal cDNAs and genes and identified the key amino acid residues that were necessary to take 10,000 keratin subunits and put them together and get a single one of these 10 nanometer filaments that extend inside cells all across from one side to the other. I learned that the mice and then subsequently humans that are mutant for these keratins have a blistering skin disorder and skin barrier defects. And so the initial focus that I had was on keratin 5 and keratin 14. Those were the first keratins that we had cultured. And we worked out the genetic basis then of this blistering skin disorder, which was epidermolysis bullosa simplex, a blistering disorder where the stem cells that are the innermost layer of the epidermis turned out to not be able to withstand the mechanical stress, and therefore, upon mechanical stress, would break and rupture, and that caused a blister. This gave us new insights into the function of the cytoskeletal network, which at the time had not been studied. And it allowed us also to identify other structural proteins and their associated human genetic disorders. And so we now know that there are more than 30 different human disorders that involve defects in intermediate filaments or their associated proteins. We also know that there are over 100 different intermediate filament proteins expressed in all cells of the body, and their antibodies have been broadly used in diagnostics for hundreds of diseases. So if we fast forward to recent times, what we know is that it's now possible to do whole body regenerative medicine from disease-corrected epidermal stem cells. All that's necessary is to take small bits of cells, grow them into culture, and correct those cells' genetic defect. And in this case, one of the most severe types of blistering, called junctional epidermolysis bullosa, the work that was done by Michaela DeLuca, another former postdoc in Howard Green's laboratory, was able to correct the mutant cultured stem cells. One can now do this by introducing the missing gene and gene therapy, or by correcting the mutant gene in CRISPR-Cas. And this shows you that same child that I showed you before after treatment. So these types of approaches that were initially based on the genetic basis of disease, that was some of the work that my laboratory had done, but now applied in a clinical setting, have turned out to really make a difference for these, what used to be absolutely devastating disorders. So what lessons have we learned about stem cells from burn operations? We learned that in burn operations, healthy epidermis is removed from the patient and either expanded in culture or grafted directly. And this engrafted skin never made sweat or hair. So that right away tells you that there are either other types of stem cells that exist within the skin responsible for generating these tissue structures, or that there are various different niche components that differ between the stem cells of the epidermis and those of the hair follicle and these other appendages. By contrast, despite complete removal of the healthy epidermis for engraftment, the denuded skin generates the epidermis in full and continues to make sweat and hair. And that tells you that either hair follicle and or sweat gland stem cells are capable, when challenged with the wound setting, to be able to undergo fate changes to essentially repair the epidermis. So we learned quite a bit from these early studies, and my laboratory went on to really understand at a molecular level, what do these differences mean? And we showed that there were different niches for the skin stem cells. We showed that the communication really determines what types of adult skin stem cells are going to be, what they're going to do, and when they're going to do it. And we now know that the epidermal stem cells are in the innermost layer of the skin epithelium. We know that there are other stem cells responsible for the base, the bulk of the hair follicle, the upper part of the hair follicle, and we know that there are stem cells that resist in an anatomical region known as the bulge, and those stem cells are responsible for the cyclical activity that we see in regenerating the hair follicle and producing hair. We also know that the niches endow these stem cells with unique gene expression and tasks. So normally the hair follicle stem cells will only make hair, but it's when they're challenged in a wound setting that they're capable of undergoing these fate changes. We also know that epidermal stem cells fuel epidermal differentiation. They produce the skin's barrier, and they produce antimicrobial peptides to be able to fight off infections. We know that the hair follicle stem cells fuel the hair cycle in which a new hair follicle and hair are formed. And finally, as I mentioned, we know that upon injury, hair follicle stem cells can regenerate the epidermis, and thereafter, once they've done that, they act like epidermal stem cells. And so, what are the lessons that we've learned in studying stem cells? We learned that stem cells are architects of their own niches. The skin stem cells began as one homogeneous monolayer of keratin 5, keratin 14 expressing cells, but then it's their communication with the other cell types that populate the skin that create these niches. The communication between the niche and the stem cells is a two-way street and dictates stem cell behavior. And finally, to change this behavior, stem cells must receive signals from their niche microenvironment or from the microenvironment itself. So what happens when the environment changes? Well, all it takes is a look at the Galapagos Islands to give us an indication of how skin changes over evolution. And there's tremendous numbers of changes that depend upon and have evolved as a consequence of the various different extremes of our environment. But what happens when our climate changes faster than ever before, as it currently is doing? We know that plants are altering the pollens and allergens that they generate. The carbon emissions and air pollutants are becoming more complex and numerous. We know that the ozone layer is thinning, that existing pathogens and allergens and microbes are shifting their territory. We're now seeing dengue virus in Texas, which we never saw before. There are new pathogens, allergens, microbes, and pollutants that are emerging, as is painfully evident by the recent pandemic. And finally, bearing the brunt of these environmental changes is the skin epithelium. It is the barrier and our body's interface with the outside world. So the skin epithelium is subjected to not only mechanical stress, ultraviolet stress, oncogenic stress, wounds, microbes, metabolic stress, noxious agents, temperature and pH extremes. If you go outside on a warm day and you come back inside, your skin is a very different temperature than the rest of your body. Your rest of your body maintains 98.6 degrees Fahrenheit, but the skin is very different and adjusts with temperature. pH, the pH of the skin is 5, highly acidic, yet the stem cells reside tucked away in a nice pH 7.4 environment. And so really for the stem cells to be able to adjust to all these different changes in the microenvironment and still survive and produce epidermis, there has to be a mechanism for the stem cells to do that. And we've been very interested in understanding that. We've learned over the years that if epithelial stem cells malfunction, either in making a proper barrier or in communicating with the immune system upon a barrier breach, then chronic inflammation and increased susceptibility to cancer can occur. We've learned that mutations that cause defects in the skin's barrier enhance inflammation. So I already mentioned to you the skin blistering disorder that arises when keratin 5 and 14 are mutated. But when keratin 1 and keratin 10 are mutated, those are the keratins that are produced in the outermost layers of the skin, then you see quite a different disorder. We see crusting at the flexural joints, indicating a mechanical problem, but we see a hyperthickening of the skin. And the reason for that is that these patients also show increased susceptibility to inflammation and skin cancer because there are two complicating factors. First, the defective barrier is prone to infection, and that's going to lead to hyperproliferation and chronic inflammation. But secondly, the stem cells are trying to rescue that barrier. They're making more and more layers, trying to produce a barrier. It's a futile rescue in the case of keratin mutations, but in turn, this leads to hyperproliferation and increased cancer susceptibility. We looked at another disorder, atopic dermatitis, which affects up to 15% of the world population. Most of these patients are placed on immunosuppressive drugs. There are environmental contributors, and there are genetic determinants. But instead of just being exclusive for interleukins, for instance, involved in inflammatory responses, researchers found filaggrin, a large protein found in these green granules of the outer skin layers, to be mutated. So for a long time, people just dispensed with this, saying it's a large gene, there are going to be mutations or polymorphic variations that are seen. But as more and more cases of atopic dermatitis cropped up that had mutations in filaggrin, people began to gain more and more interest. Felipe Garcia Carroz, when he was in my laboratory, focused on this. And what we learned is that when filaggrin is defective in patients, the granules don't form properly and the outer surface layers don't flatten and the barrier is weakened. And it turns out to be a process driven by liquid phase transitions. So in 2023, the World Health Organization ranked chronic inflammatory disease as the greatest threat to human health. There are many risk factors: mechanical stress, pollution, allergens, chemical irritants, aging, obesity, smoking, Western diets. And these are common disorders. They include not only psoriasis and atopic dermatitis and chronic wound healing, but also bladder infections, asthma, inflammatory bowel disease. And there are several common properties. They're all triggered by an irritant. Could be a chemical, an infection, a mechanical trauma. The inflammation comes and goes, and it often recurs, sometimes years later, in the exact same spot as before. And right away, that tells you that the skin is retaining some kind of memory that it's seen inflammation before, sometimes years earlier. Recurrence is also accompanied by increased severity, and the secondary bout can be triggered by a different irritant than the first one. And so in 2015, a graduate student then in my laboratory, Samantha Larson, and Dr. Shruti Nayak, who is now running her own laboratory at Mount Sinai University, wanted to know what was causing the skin to remember this prior inflammatory experience. What was it about this memory? So they did a very simple experiment. They simply topically applied an irritant called imicumod to one side of a mouse's back. For those of you who are immunologists, this generates the Th17 response. 6 days later, the imicumod had caused the skin to become inflamed, typified by increased epidermal thickness and proliferation, similar to that of psoriasis, which is a Th17 inflammatory disorder. So 30 days after the imiquimod had been removed, the skin pathology returned to normal. Everything looked normal. There were no longer any inflammatory signs, and the skin thickness was normal. But when they gave a small wound to each side of the mouse's back, it was only the side that had previously been exposed to imiquimod that always healed faster than the other side that was naive and had not been exposed. Inflammatory memory lacks the specificity of an adaptive immune response, the B and T cells. In B and T cells, you have a particular pathogen or microbe. The B or T cells will rearrange their receptors, and thereafter they recognize the exact same pathogen or microbe. But in this case, you could treat the skin with imiquimod, with MC903, a Th2 responder, with tumor promoting agent, with a wound, with fungus, with yeast infection. And in all cases, when you let the skin resolve and then wound it, the wound still healed faster the next time around. So it didn't seem to be that the secondary stimulus was, it had to be the same as the first one. And indeed, when we repeated the experiment on the background of a RAG2 null mouse, we found that lo and behold, the RAG2 null mice, which lack B and T cells, still store a permanent memory of a bacterial or viral infection. And when these mice are pre-exposed to inflammation, their later wounds still healed faster the next time around. So we then were interested in the possibility that maybe macrophages are involved. We knew from the work of Ruslan Metsitov at Yale that following withdrawal of an inflammatory stimulus, that macrophages retain a memory of the experience called trained immunity. So you can take circulating macrophages, you can put them into culture, and their genetic chromatin changes or returns back to normal at different rates. The problem with this is that the macrophages only survive for a few weeks in circulation. Whereas in chronic inflammatory disease, we were looking for something to explain a heightened response to inflammation that could manifest months to years later. And indeed, when we removed the macrophages following an inflammatory encounter, what we found is that that did not compromise the ability of the subsequent wounds to heal faster. We also did the same for the Th17 producing cells, and we showed that while they're necessary to mount the inflammatory response, they weren't necessary to be able to harbor or sustain the memory, nor were they necessary to recall the memory. And that really led us to start to wonder whether the epidermal stem cells themselves might be harboring memories of their inflammatory experiences. And this turned out to be the case. So at the time, memory was something that was thought to be either characteristic of the brain or characteristic of the immune system. We showed that the epidermis is nearly normal after inflammation resolves, that the skin looks normal, that the pathology looks normal, that the skin proteins look normal. The skin mRNAs are largely normal. But when we looked inside the nucleus, what we found were hundreds, even thousands, of epigenetic changes, accessibility in chromatin that wasn't there before the inflammation, but lingered up to 6 months later in our mice. That's 5 to 6 human years. So now we're getting to the kinetics that resembled that of what we were looking for with regards to chronic inflammatory disease. And so this shows you an example, several examples of the early studies that we did 6 days after applying imiquimod. In the normal, the gray is the control. In the red, the imiquimod treated skin. There's many, tens of thousands of different peaks that open up. It's not surprising because the skin at this stage is hyperproliferative. All sorts of different changes are happening. But 30 days later, when the pathology has gone back to normal, what we find is we can still detect certain peaks that exist in an open chromatin state. And even 6 months later, we can see that some of those peaks remain open. We can show that these are not simply open chromatin peaks with no function, But if we cut those peaks out and we use those as enhancers to drive EGFP in mice, what we find is that in the absence of inflammation, there's no activity, as you can see on the top panel of the skin. But now if you apply imicomode to the skin, the transgene turns on, indicating that we're dealing here with inflammatory sensors that are sitting in the chromatin, usually in the enhancer regions of genes. And this is occurring long after the inflammation has subsided. The genes that are associated with these sensors are rapidly reactivated upon a secondary assault, such as a wound. Now here, it's sometimes hard to determine which enhancer goes with which gene. We're currently still doing additional studies to try to understand exactly which genes are rapidly reactivated. But we know that a cohort of them that we could identify are indeed activated upon a secondary assault. So Samantha Larson was now joined by Raj Sajath and Chris Cowley, two additional graduate students in my laboratory, who wanted to know how is memory established, how is memory retained, and how is memory recalled, again focusing on a Th17-induced inflammation in the skin. And I'll just summarize for you in cartoon fashion what we learned from doing many, many experiments at the high throughput chromatin level using a variety of different tools, including histone modification mapping, including mapping of transcription factor binding, including accessibility of chromatin, RNA sequencing. So in the closed chromatin state, where there's no inflammation, it's a naive region of the genome, what we find is nucleosomes bound and nothing is happening. But now in response to the imicomode irritant, what we find is that a transcription factor, phospho-STAT3, is induced by imicomode, and it binds to the nucleosomes of the epidermal stem cells, and it opens the chromatin of an inflammatory response regulator. But this turns out not to be enough. There's an additional transcription factor that's necessary to open the chromatin, or essentially to keep the chromatin open in an established memory state. And that additional transcription factor is FOSN-JUN. FOSN-JUN doesn't bind and open naive chromatin, but what it does do is that it allows for nucleosome modifications and also to recruit the RNA polymerase machinery that then allows for transcription of the inflammation response gene. So these two factors are essential. If we knock them out, we cannot get these memory domains established in the genome. So we also then learned that while the chromatin is open, there are stem cell factors that were there all the time but never had access to the DNA. Now they do, that the chromatin is open, so they come in and they too bind to these regions along with the histone modifications. And so now once you have the inflammation subsiding, what you find is that STAT3 is no longer there, FOSN-JUN are no longer there, but now the chromatin is kept open, presumably by these stem cell factors and nucleosome modifications. They now can exist and still bind to the chromatin in the memory state, even though transcription of the genes are down. If we knock out STAT3 at this point in time, it's not required for anything. So once the chromatin is open, STAT3 is dispensable, as we went on to show by genetics. But FOSN-JUN, on the other hand, are needed because in order to be able to activate these memory domains in response to, say, a wound, you have to bring back RNA polymerase, and it appears that FOSN-JUN do that. So even though we can dispense with pSTAT3, we cannot dispense with FOS. But keep in mind that FOS is activated in response to all sorts of different kinds of stresses. So now you begin to understand the nuts and bolts of inflammatory memory. There's a primary stimulus that is some particular transcription factor necessary to bind to closed chromatin and open up the chromatin. There then have to be stress factors, Fuchs and Jun, or AP1-like factors, in order to bind and remodel the chromatin and activate these gene regions. During that period of time, stem cell factors bind, nucleosomes are modified, and then once the inflammation has subsided and there's no longer phospho-STAT 3 and Fos, now you have some chromatin histone modifiers lost, but still there are others along with stem cell factors that remain open, and that keeps the chromatin in an open, poised state, ready now to be rapidly activated upon a secondary stimulus. And that secondary stimulus can be any sort of trauma to the skin, because many different things activate Fos. Fos is basically a general stress-induced factor. So what do we now know about memory? Is it just the skin? It turns out that airway epithelia, that intestinal stem cells, the blood-producing stem and progenitor cells all harbor certain kinds of inflammatory memory that is very similar to what I just described to you. Fos and Jun is really a universal mediator of inflammatory memory. We analyzed a whole variety of different databases that had been published before and utilized our algorithms and our analyses to be able to look at the parallels between the memory that we had uncovered and these other situations that involved inflammation. And it turned out that even though STAT3 often differed, it could be STAT1, it could be NF-kappa B, different kinds of inflammatory stimuli, transcription factors, that at the front end and back end of memory was always AP1 or Fos and Jun. And so the molecular mechanisms of epidermal stem cell memory bear strong resemblance to the way in which our neurons store and recall memories in the brain. If you recall the work of Eric Kandel, a Nobel laureate and prominent immunologist, AP1 turned out to be an important factor in establishing and recalling memories. It really raises the question of whether all tissues of our body might have the ability to learn from their experiences. Why not? And especially over evolution, that would be a good, beneficial thing. And can one tissue pass on its experiences to the other? That's an interesting question as well that hasn't been studied to the same extent, but one that is broadly of interest, I think, with regards to tissue and whole body fitness. And indeed, that question bears to mind the studies of Yasmin Belkade and her colleagues, who several years ago showed that if you feed a pregnant mouse a pathogen, that pathogen goes into the intestine and creates inflammation in the intestine. There's a systemic component in that case that then goes to the fetus and creates inflammation in the fetus, even though the fetus has never seen the pathogen. That then gets passed on to the offspring, so that once the mice are born, now you end up with a predisposition of the stem cells in the intestine to inflammation, as well as protective immunity to other types of pathogens. And again, the pathogens need not be the same as the initial pathogen. And so these data really, I think, open up the door for many different questions in the future. And in fact, it's been known since the 1930s that infants that are vaccinated against one bacterium develop protection against another. It's been known that plants that survive one pathogen are often resistant to other pathogens. It's been known from our work that epigenetic memory really affords a means to apply the lessons learned in becoming sensitized by one stimulus towards encounters with a brand new stimulus. And as I've shown you, it can have both beneficial, broader pathogen resistance, for instance, and deleterious chronic inflammation consequences. So what about beyond inflammation? What happens in wound repair? And do stem cells remember a wound experience? So here we were particularly interested going back to those early studies that I described to you about Howard Green's pioneering work on skin culture engraftments and on skin engraftments. And the natural skin that is engrafted in a burn operation the area that was removed, where the epidermis was removed, can regenerate the hair. And basically the hair follicle stem cells could do that. So we basically generated the same kinds of wounds as exists in burn engraftment experiments, only this time we did it on a mouse. And when we did that, we found that the hair follicle stem cells are indeed mobilized. They move up and they can repair the epidermis. And thereafter, in their new niche, those cells can behave as epidermal stem cells. So this was the perfect example. It allowed us to look at the stem cells, excuse me, during the period of when they got a wound, they exited their niche, they migrated up into the wound bed, they underwent a fate change to become epidermal stem cells, and they repaired the missing epidermis. We asked, do they have memory, and are they accumulating different memories of these experiences all along the way? And it turned out that they are. So we found that one of the memories is the quicker response to secondary stimuli. Another memory was the increased ability to migrate and heal subsequent wounds. The stem cells, when they're migrating into the wound bed, remember that they migrated, and they're faster at migration in the next time. They also acquire an increased plasticity. They were able to make epidermis, but now that they're epidermal stem cells, they can actually be forced to make hair and they do so much better than a normal naive epidermal stem cell can. And when you think about all three of these different consequences and memories that I've described to you, these are beneficial to wound repair, but they are constitutive hallmarks of cancer stem cells. And so in this regard, what we know is that prior inflammation often heightens the risk of cancer. We know that patients with psoriasis and atopic dermatitis have an increased risk in cancer. And we know that from the work of the Viale lab and Low lab, that if the pancreatic epithelium gets an inflammatory experience, that it can develop an inflammatory epithelial memory. And that basically allows it, when acquiring a mutant oncogenic RAS gene, to progress to pancreatic cancer in a way that a naive pancreas doesn't do. There are other features of a wound response with relevance to cancer. In the work of Siqi Liu and Yunha He, two former postdocs in my laboratory, we showed that when blood vessels are broken, the tissue is deprived of oxygen. The skin stem cells at the wound edge turn on and secrete interleukin-24 in response to hypoxia. That then activates interleukin receptors, but those interleukin receptors are on the surface of blood vessels, fibroblasts, epidermal cells, immune cells. They all express the receptors. And so when this happens in an injury, what happens is that even though interleukin-24 is made by the epidermal stem cells at the wound edge, those stem cells are orchestrating the whole wound repair process. They're bringing in blood vessels, angiogenesis, fibroblasts, immune cells, all to mobilize the repair process. So this has really given us new insights and really tells us that the stem cells of our tissues are absolutely critical, not simply in maintaining their tissue, but really in orchestrating responses to a whole variety of different challenges. And with regards to that, there are many questions and challenges that remain. We want to know, how long do memories last in tissues? We want to know what happens to our tissue memories as we get older. In a world where we're facing an ever-changing environment, are we going to be able to erase the bad memories and keep the good ones? And finally, can we use our knowledge of wound repair to develop new therapeutics for cancers and other chronic inflammatory conditions? And so I started with skin, and I'll end with skin. We're dealing with stem cells that are coping with the continual changes in the environment And that problem is only going to be more important as we move forward in today's world. It's all about survival of the fittest. How do the stem cells react to all these different experiences and do their best to still do their job and do their job efficiently? So often when you're looking for something, it's very hard to find it. It's really when you're not looking for it that the answers come. And that certainly was the case with the story that I presented to you today, and in fact, with my life history as a research scientist. So I'll finish then with a few more photographs from my repertoire. And whether we're young or whether we're older or getting older, I think there's still an endless fascination with our skin. And this area of research is going to keep us busy for some time to come. So these are the people in my laboratory responsible for the work that I talked about today. I have a lab that's pretty well balanced between students and postdocs. And in addition, we have the MD, PhD clinical scholars. We receive our funding not only from the Howard Hughes Medical Institute, but also from the National Institute of Health, and also from the Sanofi Foundation, who has been supporting work that I didn't have a chance to talk to you about today, but dealing with what are we going to do about emerging pathogens, and how does the skin cope with these various different assaults. So our main collaborators are Dana Pear, Gabriel Victora, Daniel Musida, and Charlie Rudin, and I'd be happy to take questions. Thanks.
Thank you so much, Elaine. This was really one of the most inspirational lectures that I have heard in a while. I think I do have some questions, but let me just quickly introduce the next week's speaker. So this is a bioengineer, Gilda Barabino, who is the president of the All-In College of Engineering, and she will speak broadly about the tissue engineering for human health. So maybe to start with the last topic that you cover, which is the inflammatory memory. I mean, this seems to be opening door like to a whole new field. It's very broad. It seems to have like an potential that we cannot even capture at this point. So I wonder, how much do you know at this point about, besides macrophages, which other cells own this feature? So how much do you know about where it comes from? Are these immune effects of, say, macrophages on epithelium or the other way around? Like, what is the scope that you can see at this point based on the studies that you have already completed?
Yeah, so what we're finding, and I think, it's a long time that we kept on focusing on the immune cells, thinking that the immune cells must be the ones that are responsible for this long-term memory. And it was really by process of elimination that we finally realized the stem cells are actually there for the long haul too, and maybe they could be harboring memory. I think we now know that there are many different stem cells that have similar kinds of memories. I think they haven't been mechanistically studied maybe to the same depth, but all the parallels are there. And I think the parallels, again, to learning and memory in the brain will be very interesting to look at in the future. There have been new studies that, in addition to the stem cells that I described, new studies that suggest that that neurons can respond to inflammatory stimuli and basically harbor or acquire different kinds of memories in the brain. So I think that's going to be a very exciting area in the future. And I think this general issue that we're focused on now is are there different flavors of memory? We know from our skin wounding studies that stem cells, it's not just about a Th17 response, the stem cells can respond to all sorts of different kinds of experiences. And I think it'll be interesting to see whether there are multiple different kinds of inflammatory memories. Do they all converge on the same mechanism? Do they converge on the same set of genes? Is it a small cohort of genes that they're converging on? Is it a broader, are there different kinds of flavors there? So I think there's still a lot to do. We're focusing on many different aspects of epigenetic memory, and really also drilling in to try to understand deeper into the underpinnings of memory.
Yeah, I mean, it's also related, obvious related question is based on the fact that it's hard to find a process in our body that is not affected by the immune system, either innate or adaptive. So do you think that it is like inflammation is central to the process and then it affects other cell types? Or do you think that there may be a very direct effect to, say, epithelium or other cells that sort of doesn't go through the inflammatory path?
Yeah, well, we know in wounding that when you remove the epidermis, the hair follicle stem cells have to move into the wound bed and acquire inflammation, an inflammatory experience. And so the stem cells that are back in their niche that now migrate outward and repair the epidermis actually have a memory that they used to be in a hair follicle stem cell niche. So that's an example where we think it's pure epithelial centric. And the other aspect is migration, where we can take the stem cells that have experienced a wound repair, we can take them out of their niche, and if we put them into culture, they will migrate faster than stem cells that have not experienced a wound. So whereas the inflammatory experience, we've shown is a separate entity. We can stimulate in the inflammatory experience by mobilizing the epidermal stem cells to repair a wound in a deep wound where there aren't any hair follicle stem cells. And they have an inflammatory memory that is very similar to the inflammatory memory made by the hair follicle stem cells that have experienced the inflammation. The difference is that they're missing that memory of their niche origin because they are epidermal stem cells. They never changed their fate. So we've been able to tease apart some of these types of memories. And I think that's one of the aspects that's so fascinating, because when you think about all the different kinds of stresses that our body is experiencing, particularly our skin, I think it's going to keep us busy for quite some time to come.
Yeah, so there is actually a question you got quite a few questions. A question from Sanjay Kurel, who says that tissue and stem cells, and including MSCs, are mechanosensitive, and they do remember their past environment. Do you think that mechanical memory and inflammatory memory could somehow coincide or even interact in the stem cells?
Yeah, I love the question. Ask me again in a few years. But I think it's a wonderful question, because certainly, especially with regards to the skin, you rub your skin and you don't expect your skin stem cells to dissolve. And if you do the same thing with your liver is in bad shape. And so here we have this mechanical infrastructure that is built up, that we studied for quite some time in the epidermal stem cells. But you have superimposed on that, what are the experiences that those stem cells have undergone? And I think it's one of the reasons why there are so many different disease-related effects of mechanical stress and the like that crop up later on in life. I mean, certainly there are consequences of degradation of the extracellular matrix. The stiffness of the skin is no longer what it used to be. as we get older. But there are many other types of mechanical stresses. And in fact, we've shown, and others have shown, that the cells, when the stem cells differentiate to produce this outer barrier surface, the barrier surface is extremely stiff. And so the barrier surface itself is able to feed back mechanical pressure to the epidermal stem cells. We've shown that the basement membrane is also can vary in different stiffnesses depending upon its composition. Same thing is true for the extracellular matrix. So let alone external trauma, there are all sorts of built-in types of trauma that are going to be changing as skin ages, for instance, that I think are going to be very interesting. So I think you could basically look at mechanical memory for a long time in and of itself and come up with a lot of interesting findings.
So few people asked the variations of essentially the same question. If you don't apply just one stimulus, but you like apply trains of stimuli, or you may vary like positive, negative, positive, negative. I mean, what prevails? Is it like precedence, like what you apply first, and then the cells remember, or you can override the first stimulus by how much do we know about it?
Again, I had to say, ask me again in a few years. You guys are asking wonderful questions. The answer is we don't yet know, but I think it's a really fascinating question to look at. A priori, you could imagine that since we know we can accumulate memories over time, that we're simply accumulating lots and lots of memories. Maybe that explains why older people suffered so much more from the COVID pandemic than younger people did. If you're accumulating inflammatory memory over a lifetime and now end up with a new pathogen that you've never seen before, but can respond and stimulate the memory, then you're dealing with some, I think, really tantalizing questions, but also ones that are really clinically important. And then the other side of the coin is maybe if you just keep getting traumatic experiences, maybe there's a dulling down of the memory. Maybe there's a tolerance that develops over time. And so it could be that over time you just simply cannot accumulate any more memories, or memory has a height and isn't lost. So all of these questions, I think, are really good questions, and we simply have to do more research to be able to really get to the bottom of those questions.
So we need to really finish now. We are at the top of the hour. We need to let you go, but I would like to ask you to please answer some more questions. We will give you contact information. They're really interesting, so we can send them to you so that people get answers, because we really ran out of time. So thank you so much again. This was really fascinating, and thanks everyone for asking all these creative questions.
Thanks, Gordana.
Sure.
Thank you all.